So we continue with part 4 of our questions on the professional licensure examination. So this will be on micro B. But just as an aside, usually you may not have enough time to cover all the course content or the textbooks on microbiology. So it's very important that you solve questions. When you solve questions, questions have a way of hiding the answers, and the process of finding the answers in itself is so revealing. You're going to learn a lot by trying to solve questions. So instead of waiting to learn everything before you solve questions, what I advise is that even if you've not finished learning or if you have very little time to prepare for your examination, a very smart approach is to actually focus on solving questions and let the questions lead you to the answers. It helps you to avoid majoring on the minor or minoring on the major. And so this is a very good approach to use. Always solve questions. Don't be afraid that, I've not covered all the knowledge. No, you may not be able to cover all the knowledge, especially with microbiology and a lot of information about microorganisms. So this is a little advice as we move through this last part. Okay, so we continue with question 76. It says that in the disc diffusion susceptibility testing, wow, as an antimicrobial agent diffuses away from the disc, the concentration of antibiotic is A, increased, B, decreased, C, unchanged, and D, inoculum dependent. So the answer here is decreased. So this question is basically saying that when you have plated and you have put your antimicrobial disc on the plate, it's saying that as the antimicrobial antimicrobial agent diffuses into the exist, what will happen to the concentration of the antibiotic? It's a simple diffusion question where there is movement from a high concentration level to a lower concentration level. So that is a straightforward diffusion concept question. So as the antimicrobial agent diffuses away from the disc, the concentration will decrease. Okay, so that will be the answer. Then 77, an aspirate of a deep wound was plated on blood agar plate and inoculated anaerobically and aerobically. At 24 hours, there was growth on both plates. This indicates that the organism is A, non-fermenter, B, obligate anaerobe, C, aerobe, and then D, facultative anaerobe. So the answer for this one is facultative anaerobe. Okay, so very straightforward. So facultative anaerobes are able to grow in the presence of oxygen or in the absence of oxygen. So the organism growing after 24 hours, both in these conditions, shows that it's a facultative anaerobe. Okay. 78. The expected colony count in suprapubic urine from a healthy individual is A, 0 colony forming units per mil, B, 100 colony forming units per mil, C, 1,000 colony forming units per mil, and then D, 100,000 colony forming units per mil. The answer to this one is 0 colony forming units per mil. Okay, so what's the reason? So suprapubic urine is actually urine that was collected directly from the bladder, and this urine, usually if it's from a healthy person, it should contain no bacteria, because it's supposed to be sterile. And if you compare it to urine that was collected by allowing the patient to pass to urinate the normal or the natural way of urinating, this type of urine actually bypasses the urethra, and so it's not, or it does not experience the usual contamination that you experience from the other type of urine. So that's suprapubic urine collected directly from the bladder. And then with this urine, we expect that it will have zero colony forming units per mil because it's supposed to be sterile. So that's the answer for question 78. I hope you get the explanation. Okay, then 79. It says that a sheep agar plate inoculated with 0.001 mils of urine grows 70 colonies of Staphylococcus aureus. How many colony forming units per mils of urine should be reported? A. 70 colony forming units per mil. B. 700 colony forming units per mil. C. 7,000 colony forming units per mil. And then D. 70,000 colony forming units per mil. And the answer here is D. 70,000 colony forming units per mils. So how do you solve this? This is a simple mathematics question. To find colony forming units per mils is the colony forming units divided by the volume in mils. So here the colony forming unit is 70 and then the volume is 0.001 in mils. So 70 divided by 0.001. When you do that calculation, you are supposed to get option D. That's 70,000 colony forming units per mils. Okay, so that's the answer for 79. Let's move to 80. It says that when stool examination is negative, the preferred specimen for the diagnosis of paragoniasis is A. Bowel drainage, C. duodenal aspirate, C. sputum, and then D. rectal biopsy. So the answer here is sputum. And if you look at paragonimiasis, it's caused by Paragonimus westermani. This is a lung fluke, and it lays eggs in the lungs. And so when you are not able to detect it in stool, another sample that you can take is sputum. It will help you identify the eggs. Okay, so that's about that. Then question 81. Organisms that can be easily identified to the species level from the ova in fecal specimen include A. Metagonimus yokogawai. Then heterophiles. B. Paragonimus westermani. And then Hymenolipes nana. C. Neceta americanus and Ancylostoma duodenale. Then D. Tinea solium and then Tinea saginata. The answer here is D, which is Tinea solium and Tinea saginata. So Tinea solium is pork tapeworm and then Tinea saginata is beef tapeworm. Then these have similar morphologically looking eggs, but then they are distinguishable by certain specific characteristics that they both possess. And so this becomes one of the organisms that you can use their eggs to actually help distinguish them much more easily as compared to the option A, that is Metagonimus yokogawai and heterophiles, which is more challenging to actually distinguish them based on their ova in fecal specimen. If you look at high option and B like this, the Hymenolipes nana like this, it has eggs that are relatively small and resemble that of other tapeworm, but can be identified with specific characteristics in the egg, though it's not as easy as the Tinea species. And so the best answer here is Tinea solium and then Tinea saginata. If you look at option C2, Neceta americanus and Ancylostoma duodenale They have similar eggs, and then it's very challenging to actually differentiate them in fecal specimen. Okay, so the answer here is Tinea solium and Tinea sedentata. Question 82, it says that anaerobic infections differ from aerobic infections in which of the following? So A, they usually respond favorably with aminoglycoside therapy. B, they usually arise from exogenous sources. C, they are usually polymicrobic. And D, gram stain of specimen are less helpful in diagnosis. The correct answer here is they are usually polymicrobic. Okay, one thing about anaerobic infections is that they are polymicrobial. That is to say that they involve multiple types of microorganisms, including both anaerobic and aerobic organisms, because the natural habitat of these anaerobes also have aerobes there. If you take sites that you see anaerobes are at, let's say, the mouth, gastrointestinal, or femogenital tract, you realize that there are aerobes also there. So because of that, we say that they are usually polymicrobic. If you look at option A, they usually respond with aminoglycoside therapy. So aminoglycosides are generally ineffective against anaerobes. So instead, anaerobic infections are typically treated with antibiotic effective against anaerobic bacteria such as metronidazole and clindamycin. And so option A won't be a viable answer. Then if you look at the B, they usually arise from exogenous sources. Anaerobic infections often arise endogenously, meaning that they originate from the patient's own normal flora rather than from external sources. Then if you look at option D, gram stain of the specimen are less helpful in diagnosis. So gram stain will be still useful in this case. They are not always definitive for anaerobic because they might not always be visible or easily identified. However, they can still provide some diagnostic information. But then the correct answer here is that they are usually polymicrobic. Okay, so question 83. It says that the best procedure to differentiate Listeria monocytogenes from Conibacterium species is A, catalase, B, motility at 25 degrees, C, motility at 35 degrees, and D, gram stain. So the correct answer here is motility at 25 degrees. Yeah, so at 25 degrees, you realize that Listeria monocytogenes is motile, usually has a typical tumbling motility under the microscope, as compared to Conibacteria, which exhibit no motility. Then if you look at option A like this, it says catalase test. Catalase test is actually positive for both of the organisms, so you cannot help differentiate them. Then if you look at motility at 35 degrees Celsius, all the organisms are not motile at that temperature, so that answer is not also correct. Then option D, gram stain, it says that how do I say? It says that while the gram stain can show the general morphology of bacteria, it's not sufficient to differentiate between Listeria monocytogenes and Conibacteria species, as both of them appear as gram-positive rods. So that's about it. So the correct answer here is B. Okay, then 84. It says that all species of Pseudomonas have the enzyme to oxidize A, naphthalenamine. D, these words are difficult to pronounce. B, dimethylaminobenzoate. glucosylzaldehyde. Then C, glucopyranoside. I hope I got that one right. Then D, tetramethylparaphenylnediamine. So the answer here is D, tetramethylparaphenylnediamine. Okay, so this basically asking about the oxidase test, which is a key test for identifying Pseudomonas species. So the oxidase enzyme reacts with tetramethylparaphenylnediamine, resulting in a color change, typically from colorless to purple, indicating a positive result. So this reaction is characteristic of Pseudomonas, and then it's used to differentiate it from other gram-negative bacteria. Okay, question 85. It says that a urethral swab obtained from a man with a urethral exudate was plated directly on chocolate agar and modified Theamartin agar, and a gram stain was made. The gram stain showed gram-negative diplococci, diplococci. The culture plates were incubated at 35 degrees Celsius, but had no growth at 48 hours. The most likely failure for organism growth is that the A, wrong media were used, B, gram stain was misread, C, organism only grows at room temperature, and D, organism require carbon dioxide to grow. So the correct answer here is D, organisms require carbon dioxide to grow. Okay, so let's analyze our options. It says that the fact that it says a urethral exudate, and then it also mentions that it's gram-negative diplococci. This is indicative of Neisseria gonorrhea. This typical gonorrhea caused by Neisseria gonorrhea. And one characteristic of this organism is that it requires carbon dioxide-rich environment for optimum growth. It requires carbon dioxide, so usually we'll be incubating it in a carbon dioxide incubator, or you'll be adding carbon dioxide to the culture environment. So I think that's the answer. If you look at option A, which is that wrong media were used, both chocolate agar and modified Theamartin agar are appropriate for culturing Neisseria gonorrhea, because chocolate agar provides nutrients, and then Theamartin agar is also enriched and selective for the Neisseria species. Then if you look at option B, it says that gram stain were misread. So the presence of gram-negative diplococci is consistent with gonorrhea, with Neisseria gonorrhea. So the saying that the gram stain was misread is very unlikely. Then option C is saying that organism only grows at room temperature. That answer too is not viable, because Neisseria gonorrhea grows optimally at 35 to 37 degrees Celsius and not at room temperature. Okay. So that's about question 85. Let's move to the next question 86. Okay, so a biomedical scientist is reading a gram stain from a CSF and observe mini neutrophils and lancet-shaped gram-positive cocci. Which set of chemistry and microbiological CSF results will most likely be seen in someone with this type of infection? So A will have increase in WBC, increase in glucose, and then increase in protein. Then B will have a decrease in WBC, decrease in glucose, decrease in protein. Then C will have an increase in WBC, a decrease in glucose, and an increase in protein. And then option D, we have a decrease in WBC, increase in glucose and then a decrease in protein. So the question is asking which of these is likely. So A, objective answer A is in result A, B is in result B, C is in result C, and then D is in results D. So our answer for this question is C. So we would see an increase in WBC, a decrease in glucose, and an increase in protein. So why does that happen? Okay, so it says that there's a culture of CSF specimen. Yeah, CSF. When there's bacteria in CSF and then it's being cultured, it's because there is some it will cause some form of inflammation, and then because of the inflammatory response there will be increase in WBC, and then there will be a decrease in glucose, which is caused by the bacteria consuming the glucose. And then the inflammatory response itself can also affect the glucose level. And we say that option C because proteins too will also increase. So the protein level will increase due to the breakdown of the blood-brain barrier and also the inflammatory response. So option C is actually our best answer. We'll have an increase in WBC because of the inflammatory response, and then we'll have a decrease in glucose because the bacteria is using the glucose, and then we'll have an increase in protein because of the inflammatory response, which causes cells and cytokines and things to come into the area, and then because more importantly there's a breakdown of the blood-brain barrier. Okay, question 87. It says Vibrio cholerae can be isolated best from feces or rectal swab on A using methylene blue agar, that's EMB, hectone enteric agar, Salmonella shigella agar, that's SS agar, and then thiosulfate citrate bile salt sucrose, that's TCBS agar. And the answer here is thiosulfate citrate bile salt agar. Okay, so one thing about thiosulfate citrate bile salt sucrose agar, wow, is that it is selective and differential medium specifically designed to isolate Vibrio species, including Vibrio cholerae. So it contains bile salt to inhibit the growth of other bacteria, and then sucrose, which the Vibrio cholerae ferments to produce yellow colonies, helping differentiate it from other Vibrios that do not ferment sucrose and produce green colonies. Okay, then if you look at option A, like the eosin methylene blue agar, this is primarily used for isolating and differentiating gram-negative enteric bacteria, particularly Escherichia coli, and does not selectively isolate Vibrio cholerae. Then if you look at hectone enteric agar, this one is used for isolating enteric gram-negative bacteria, particularly Salmonella and Shigella, and it's not selective for Vibrio cholerae. Then Salmonella Shigella is selective, of course, for Salmonella and Shigella species and does not support the growth of Vibrio cholerae effectively. Okay. Next question, 88. It says that diagnosis of typhoid fever can be best confirmed by culture of A, stool, B, urine, C, blood, and then D, bone marrow. The answer here is bone marrow. Bone marrow. Okay, it's bone marrow because bone marrow is highly sensitive for the detection of Salmonella typhi. Even in cases where you have blood culture being negative, you realize that bone marrow will actually be more revealing. And so it's particularly useful for diagnosing typhoid fever in cases of prolonged illness or when the culture have failed to yield results. So blood culture is also useful. Stool culture is also useful, but then in chronic situations, people who have acute diagnosis, who have been acutely infected, you may have negative test results for the stool test. And then usually for urine B, you will not find typically the typhoid bacteria in there. So it's not so. So the best answer here is option D. Okay, question 89. Question 89. It says that most commercially available blood culture media, such as the Bactec blood culture media, contain sodium polynithosulfonate, that's SPS. So SPS is used as anticoagulant for blood culture because it A. inactivates penicillin and cephalosporin, B. prevents clamping of red blood cells, and then C. inactivate neutrophils and components of serum complement. Then D. facilitate growth of anaerobes. So the correct answer here is C. That's inactivate neutrophils and components of serum complement. So SPS. So those Bactec bottles that we've been seeing for blood culture, they contain this anticoagulant that we call SPS. And then the main function of these SPS is to prevent the blood from clotting by inhibiting the activity of certain proteins involved in the clotting process. Then additionally, it also neutralizes the effect of neutrophils and components of the serum complement system. So, which, if you look at the other options like A, inactivate penicillin and cephalosporin. So, SPS does not have this effect. The inhibition of antibiotics is not its primary function. If you take option B, it says prevent clamping of red blood cell. So SPS does not prevent clamping of red blood cell. It only serves as an anticoagulant. Then if you look at option D, facilitate growth of anaerobes. So SPS does not specifically facilitate growth of anaerobes. Its primary role is related to anticoagulation and inactivating of certain immune response. So that's about that. The correct answer here is C. Okay, then question 90. Question 90. It says that the concentration method for diagnosis of blood parasites include all the following except A. cellophane tape preparation, B. microhematocrit centrifugation technique, that's the Wu method, and then C. triple centrifugation technique, and then D. the knot concentration technique. So the answer here is A. cellophane tape preparation. Cellophane tape preparation. So let's go through the answers one by one. If you take option B like this, the microhematocrit centrifugation technique or the Wu method. So this technique is used to concentrate blood parasites, particularly for diagnosing malaria. So it involves centrifugation or centrifuging blood to separate the components and making it easier to detect the parasite. So that's the Wu method. Then if you take the option C, triple centrifugation technique. So this method involves multiple centrifugation steps to concentrate and isolate blood parasites, such as malaria parasite, from a blood sample. Then the knot concentration method so primarily used to concentrate and detect microfilaria in blood samples. So it involves mixing the blood with lysine agent and centrifuging to concentrate the parasite. But then if you look at the cellulose tape preparation, that's option A, which we are selecting to be our answer here. This method is not used for diagnosing blood parasite. It's typically used for detection of pinworm in perianal area, like in the perianal area. So that's what this method is used for. It's for pinworm. So that's our answer for question 90. Then question 91. Was it where a patient's urine has a chylous appearance? What kind of parasite may be suspected? A. Schistosoma hematobium. B. Onchocerca volvulus. C. Ruchuria bancrofti. And D. Leishmania donovani. So the correct answer here is C. Ruchuria bancrofti. So Ruchuria bancrofti is what causes lymphatic filariasis, or commonly known as the elephantiasis. And then when that happens, there is a lymphatic obstruction, and so the lymph will actually leak into the urine, producing this chylous, or if you want, milky appearance in urine. So it's this bad guy that does that. That's Ruchuria bancrofti. Okay, if you look at schistosome causes urinary schistosomiasis, and it causes blood in urine. That's hematuria. So it's not associated with chylous appearance in urine. Then if you look at onco causes river blindness. That's onchocerciasis, and then it's primarily affects the skin and then the eyes. It does not cause any chylous urine. Yeah, it doesn't. Then if you look at Leishmania donovani, so this parasite causes visceral leishmaniasis, which affect the liver, spleen, and bone marrow, and it's not associated with chylous urine. Okay, so our answer here is Ruchuria bancrofti. That's our answer. Question 92. It says that what is the likely organism responsible for this condition? A. Giardia lambia. B. Cryptosporidium parvum. C. Chylomastix menili. Hey, these organisms. Then Entaviva histolytica. You see, question 92, I think it's not complete. Something was left out, maybe a picture or something like that. And this reveals to you that the questions are taken from a question bank, so they select them. And then I think most likely the person selecting this did not pick up the picture. So that's why solving these questions are very, 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 very important. Most of them will come or would will come in the licensure exams once again. So it's good to go through them. Okay, so with that having said that, let's move to the next question. Question 93. It says that in this diffusion susceptibility test, which of the following can result if the disc is placed on inoculated media and left at room temperature for an hour before inoculation? So it's saying when you delay the inoculation, after you have placed your antibiotics on it, what will happen? So A says that the antibiotic will not diffuse into the media, resulting in no zone. Then B, zone of smaller diameter will result. C, zone of larger diameter will result. And then D, there will be no effect on the final zone diameter. Okay, so the answer I chose here with my reason is option B, zone of smaller diameter. And the reason is that with this Kevibar method, it's crucial that the antibiotic disc is placed on the inoculated agar plate and then inoculated promptly. If the disc is left at room temperature for an extended period, the antibiotic will begin to diffuse into the medium prematurely and unevenly. This can lead to a reduced concentration of the antibiotic at the disc edge, so which will then now result into a smaller zone of inhibition when the plate is finally incubated. Good. So I think that is the reason the antibiotic will actually lose some of its concentration, and so when the bacteria is actually grown and then we need it, it won't be available in the quantity that we need, which will result in a smaller zone of a smaller diameter. Okay, so that's option B. So that's the answer for question what do you call it? Question 90. 3. Okay, so let's move on. Question 94. Most commercially available blood culture media, such as the Bactec blood culture media, contain sodium polyanitosulfonate. It's worse. So SPS, let me just say SPS and go my way. SPS is used as anticoagulant for blood culture because it A. inactivate penicillin and cephalosporin, B. prevent clamping of red blood cells, C. inactivate neutrophils and components of the serum complement, and then D. facilitate growth of anaerobes. If you are paying attention, this question we've solved it already, and they repeated it again. So another evidence that they take questions from the question bank and then they repeat them. Most likely you're going to see it in your exam somewhere, somehow. Yeah. So we've solved that again. The answer is C. So let's not waste time on that. 95. So which of the following method is inadequate for the detection of Staphylococcus aureus strain, which reduces susceptibility to vancomycin? That's VISA. That's vancomycin intermediate Staphylococcus aureus. So the options here are broth microdilution, then B. agar dilution, C. gradient diffusion, and then D. disc diffusion. The correct answer here is disc diffusion. 96. It says that an antibiotic used to suppress or kill contaminating fungi in media is A. penicillin, B. cyclohexidine, B. streptomycin, and then D. amphotericin B. The correct answer here is cyclohexidine. Cyclohexamine. Okay, cyclohexamine. So why? So cyclohexamine is commonly used as a microbial media to inhibit the growth of fungi such as yeast and mold while allowing the growth of bacteria. So it's particularly useful to select media for isolating and identify bacteria where fungal contamination might be a problem. If we take option A, penicillin, it's an antibiotic that attacks the bacteria cell wall synthesis and it's used to inhibit or kill gram-positive bacteria and not fungi. If you look at streptomycin, so streptomycin is also an antibiotic used to treat bacterial infection, particularly gram-negative bacteria, and does not target fungi. If you look at amphotericin B, so it's also an antifungal fine used to treat fungal infection in clinical setting, but it's not typically used to suppress fungi in microbiological media. So the correct answer here is cyclohexamide. Okay, let's continue. Question 97. It says that quality control results for this diffusion susceptibility test yield the following result: aminoglycoside zone too small and penicillin zone too large. This is most likely due to A. inoculum being too heavy, B. inoculum being too light, C. pH of molar-Hinton agar being too low, and then D. calcium and magnesium concentration in the agar being too high. So the correct answer here is A. the inoculum being too heavy. So if the inoculum is too heavy, it means that there is too many bacteria on the agar plate. This high bacterial density can lead to a smaller zone of inhibition for antibiotics like aminoglycosides because they have a higher bacterial concentration. Because the higher bacterial concentration can neutralize or reduce the effective concentration of the antibiotic around this. But if you look at antibiotics like penicillin, the larger bacterial load may result in larger inhibitory zone as the antibiotics is able to inhibit the bacterial growth more effectively as compared to a lighter inoculum. Okay, so that's about that. That's the answer for option A. If you look at option B, it says inoculum too light. So An inoculum that is too light generally leads to a larger zone of inhibition for most antibiotics because the bacterial density is low, and the antibiotics effect on it is now pronounced. Now, if you look at pH of molar-hinted agar being too low, it says that the pH of the agar usually affects the activity of some antibiotics, but it would generally cause a change in the zone size in a more uniform manner rather than producing such a specific pattern. Then calcium and magnesium concentration in the agar being too high, that's option D. So the high level of calcium and magnesium in the agar can affect the activity of certain antibiotics, particularly the aminoglycan. However, this typically affects the results more broadly rather than producing this particular pattern described. So our answer here is A, inoculum being too heavy. Okay, so 98. Indoor production in bacteriological cultures containing E. coli is made possible by the production of all the following except A, tryptophan, B, ammonia, C, indole, and then D, hydrogen sulfide. So the correct answer here is D, hydrogen sulfide. Why is the answer hydrogen? If you look at our options, if you look at our options, tryptophan. So E. coli produces indole from tryptophan through the action of the enzyme tryptophanase. So the enzyme breaks down the tryptophan into indole, ammonia, and the pyruvic acid. Then if you look at ammonia itself, ammonia is a byproduct of the breakdown of tryptophan by tryptophanase. So it's not directly involved in the production of indole, but it's a result of the process. Then if you look at indole is a product of the reaction where tryptophan is converted by tryptophanase. So the presence of the indole is what is detected in the indole production test. Then if you look at our answer, which we say is hydrogen sulfide, they say that hydrogen sulfide is produced by other bacterial processes such as the reduction of sulfates and thiosulfates, and is not involved in production of indole. It's typically detected using different tests such as the sulfide indole motility test, but does not play a role in indole production itself. So that would be our answer. So question 99, it says that all the following parameters need to be determined in describing bacterial morphology on culture plates except A, margin of colony, B, pigment production, C, elevation of colonies, and then D, fractionation of colonies. So the correct answer here is fractionation of colonies. The reason being that if you look at option A, the margin of colony, this refers to the edge of the bacterial colony and can help in identifying the bacteria. So is the edge smooth, is it irregular, or is it serrated? This is very important in identifying bacteria colony. Then pigment production. So this describes any color produced by the bacteria, which can be an important characteristic for identification. So the color that it produces usually is used for identification. Then elevation of the colonies. So this describes the shape of the colonies in terms of their height above the agar surface. Is it flat, is it raised, or is it convex? So this parameter is also very much used. However, fractionation of colonies, this is not a standard parameter for describing bacterial morphology. So that would be our answer. Okay, finally question 100, and then we are done. Woo, what a journey. Wow. If you've come to this far, like kudos. That's a lot of hard work already. Okay, so the following are examples of biochemical media commonly used in medical microbiology laboratory except A, citrate agar, B, Kliegler ion agar, C, urea agar, and D, phenytoin enteric agar. The answer here is phenytoin enteric agar, because A, which says citrate. So citrate, this medium is used to test whether an organism can isolate citrate as its sole carbon source. So it's used. So a positive test is indicated by a color change due to the production of the alkaline compound. Then if you take option B, clear ion agar. This medium is also used to differentiate between bacteria based on their ability to ferment glucose or lactose and to produce hydrogen sulfide. So it contains phenol red as a pH indicator. So you also see this one. Then urea agar. This medium is also used to test organisms' ability to hydrolyze urea in ammonia and carbon dioxide, which increases the pH and changes the color of the medium to pink. So it's used. But then if you look at our lab's oxygen, phenytoin and enteric agar. So this is not the standard or commonly used by a chemical medium. Yeah. Maybe misspelling or some confusion with another type of medium. So that's not the answer. So basically that's about that. So good luck in your exam. Bye for now.